Thanks for being here. Can you hear me okay? Thank you. Before we begin, do any of the Ocean Studies faculty have any announcements they want to make? All right, we'll get right to it then. Our speaker today is Jason Graff, and it is my great honor to introduce Jason. Jason is a friend of mine from graduate school. Jason is an associate professor of research in the Department of Botany and Plant Pathology at Oregon State University. He earned his PhD from the Graduate School of Oceanography at the University of Rhode Island in 2010 and began a postdoc position at Oregon State in 2011, transitioning into a faculty position in 2015. Jason's research interests span from viruses and bacteria to zooplankton ecology, with a strong focus on phytoplankton stocks, physiology, in-water optics, and satellite remote sensing applications. He's constantly looking to develop new methodologies and technological applications. He had a very brief career in marketing from 1998 to 2001 after graduating from the University of Illinois with a degree in advertising. He quickly found his way into an undergraduate program in oceanography at the University of Washington, where he ended up on a research vessel in the Bering Sea, and he hasn't looked back since. As I mentioned, Jason is one of my best friends, I would say, from graduate school. He and I both lived in Providence and drove a half hour to Narragansett at URI every day, so we carpooled together. And I have many stories I could tell about Jason, but I'm going to refrain because I don't want to undermine his scientific credentials right before he talks. But I'm really looking forward to this talk, and we'll listen to Jason, and then we'll do questions afterwards. So take it away, Jason.
All right, thanks, Carrie. Can you guys hear me?
Yes, we can.
Okay. All right, great. Yeah, Carrie, thank you for that nice introduction. So briefly, a quick outline of the talk today, and I really am honored to have been invited to give a talk here. It gave me a lot of time to think about sort of my history and where I've been in my research and background to where I am today. So bear with me as I walk you through some of that. So brief outline, I'll talk about my career in research and how I got where I am, and then talk about some research expeditions that I've been very fortunate to be a part of that were really large expeditions funded by NASA, the NAMES project and the Exports project, which I'll give some detail on, as well as a new project that's starting up called BioGoShip. And I'll try and walk through the logistics of some of these projects, speak towards some of the technology, specifically some of the technology that I use, and try to give some interesting, or at least what I think is interesting, research examples from my work within these projects. So how did I get here today? Carrie invited me to give a talk. I think she waited till I had a couple beers one time when I was in Maine and then sprung it on me. So here I am. But I also, looking through the faculty list in the department, I see a number of my colleagues, Leanne and Carrie, who I was also in graduate school with and know well, but also saw a number of other of your professors who have connections to University of Rhode Island. So I guess this is somewhat of an advertisement for the University of Rhode Island. So go Rams. Where I currently am, I'm at Oregon State University and part of a group we call the Phytoplankton Ecophysiology Lab, and my collaborators include Mike Barenfeld, Toby Westbury, and Alan Milligan. I currently have a master's student, Brian Verwey, and Kim Halsey in OSU microbiology. And there have been many postdocs over the years who we've had many conversations with and have helped drive a lot of ideas. And as I go through some of the work, you'll see a number of my other collaborators from these projects, which are just too many to mention. As Carrie said, my research interests really span sort of the gamut of everything from the cellular scale up to global scale applications. And I purposely move around through these scales because I'm interested in lots of different things. And if I were to focus on any one thing, I think I would get extremely bored. So I use a lot of different tools, everything from microscopy up to satellite data, and all the tools in between, the optical instruments, flow cytometers. And of course, that takes me through the span of spatial scales from cellular micron scale, things like viruses, up to global scale processes. So how did I get here? As Carrie already walked through my CV a bit, I did have a brief career in promotional marketing after getting a degree in advertising. I quickly realized that wasn't for me, and I spent about 2 years in that field and left. I still do have lots of pens with my names on them, so that's always a reminder of where I came from when I'm walking around the house. I did get a BS in Oceanography from University of Washington in 2003. I know a lot of you have senior thesis projects. Mine was on the temporal and spatial response of microzooplankton to spring bloom in Daybob Bay, which is a small bay in Puget Sound. During my time, I also volunteered for multiple cruises. My first cruise was in the Bering Sea, and that really sort of hooked me into oceanography and has sort of propelled me forward ever since. I've spent a lot of time at sea. I was calculating just before this talk. I've spent about 1.5 years at sea total. And I also spent time as an apprentice for a project at University of Washington Friday Harbor Labs. And that's really where I learned how to be part of a team and do research. And we were studying this large single-celled algae called acetabularia, and it's got a really interesting physiology and cellular structure. As a single cell, it's visible with the human eye. It gets almost as long as your thumb, so really interesting. But I really learned how to sort of take a part of a project and work as a team, and I think has sort of helped me through my career understand how to divide up work and be successful as a group. I then went on to graduate school of oceanography at University of Rhode Island, where I actually initially started doing deep sea research and worked on a project at Kikum-Jenny submarine volcano. And the far right picture at the bottom here is an image from a paper where I described a new species of polychaete worm. But following that project, I sort of realized that I really didn't want to do deep sea benthic work, and went back into marine phytoplankton and plankton in general. And so my dissertation was really a lot of method development, and on the left hand is just a schematic of a microchannel chemotaxis experiment that I designed, where we could look at the behavior of bacteria as they move towards or away from different compounds that we expose them to, specifically compounds from either competing or attracting particles. Competing bacteria are particles with attractive elements to them. But also during graduate school, I spent a lot of time doing other projects, probably why it took me a long time to get through graduate school. I worked on a long-term plankton time series and looked and analyzed the methods that were being used for that and found some faults in them, and we improved them. I worked in a phytoplankton thin layers program in Monterey Bay, and these pictures in the center at the bottom are myself and the team deploying optical instruments that profiled the water column multiple times per day, looking at different optical properties of the water, like fluorescence, backscattering, and attenuation. And then again, volunteered for multiple cruises. And that sort of really rounded out my career throughout graduate school. When I got to Oregon State University, I took a position as a postdoctoral research associate as part of a project called the NASA Phytoplankton Carbon Project. And the idea here was that someone needed to develop a method where they could directly measure phytoplankton carbon. And the idea was to use a sorting flow cytometer, and I'll go into a little more detail of that in a minute, and then where we can sort cells away from other particles in the water, and then analyze those cells for their carbon, nitrogen, and phosphorus content. And as a result of this work, NASA is currently creating a satellite product that will be available to researchers on a global scale to look at phytoplankton biomass in the surface ocean. So the schematic on the right is a diagram of the flow cytometer that I typically use. It's a sorting flow cytometer. And so a seawater sample is injected through a nozzle, and that nozzle actually vibrates at a very specific frequency so that the stream drops up or breaks up into droplets. And those droplets are analyzed with a laser, and we get different fluorescence and scattering properties. And based on those properties, we can tell the instrument to collect a specific droplet. So it adds a charge to the droplet of interest, sorts that droplet and the cell within it into a tube, which we can then use for elemental analysis. And so a lot of my work in the last 10 years has really revolved around using this instrument and applying the data from it in different ways. And I'll go into some of those examples when we get into the projects. But specifically looking at the fluorescence properties at 692 and 530 nanometers, those roughly correlate with chlorophyll, and 530 with phycoerythrin. And then the forward scatter and side scatter properties are of interest because we can relate those to properties like cell size and carbon content. So this is a picture of me at sea with this instrument. This instrument, the flow cytometer, is called the BD Influx, and we take this to sea once, occasionally twice per year. The manufacturer is not happy with us. This is about a $500,000 instrument that is designed to go in pharmaceutical labs. A number of oceanographers have adopted this instrument, and we use it quite regularly at sea, with some hiccups, of course, because when we're moving around on a vessel, instruments are not always happy. So this is an example of a global map of phytoplankton carbon that we can produce from some of the work that I did as a postdoc. And this is the data that will be soon made available to the public through the NASA biogeochemistry program. So as Carrie said, in 2015, I became an assistant professor of research at Oregon State University. And in 2021, so just last year, transitioned to associate professor. And what the professor of research really is I strictly do research, and I'm completely funded to do research. And so it's a little bit different from those who have standard positions as tenure track faculty who both teach and research. And so I spend a lot of time at sea. I spend a lot of time thinking about research, and it's really what I really love to do. And so today I'm going to go through 3 different examples of research expeditions that I've been a part of since 2015, or that started in 2015. The first one is the NASA NAMES program, and NAMES stands for the North Atlantic Aerosols and Marine Ecosystems Study. The Exports project is, whereas NAMES is interested in the surface ocean and aerosols, Exports is interested in the surface ocean and the carbon that's exported to depth. And then the BioGoShip program, I'll go into a little bit later, is a new pilot study that we're working to get biological measurements at a global scale. So the first example will be the NASA NAMES project. And this image behind us is actually a picture taken during one of the research cruises. As those of you who have been to sea know that it's not always calm and enjoyable out there. And we still have to work through these sorts of situations. So the science of names was really set out to evaluate the seasonal impacts of marine plankton on aerosols and clouds. So as phytoplankton grow and are consumed in the surface ocean, and waves break and aerosols are formed, what is the impact of those marine phytoplankton as it goes through an annual cycle of a bloom and a bust cycle? And how does that change, and how does that impact the ice clouds and the albedo of the atmosphere? And you can imagine there are a lot of different technologies that had to be used to look at this, this problem, or this interesting scientific question. So in 2014, I think the decision was made to fund the NAMES project. It's an Earth Venture suborbital project. And so NASA's interested in the Earth as well as other planets. And so they have these sort of large calls for proposals. And this was an extremely large proposal. This was approximately $30 million funded. And it's really using lots of different technologies. It's using ship-based technologies. It's using remote assets like these BioArgo floats. We actually had a C-130 aircraft that was outfitted with instruments, optical instruments that look both up and down. And this green light coming out of the plane that you see here is called a LiDAR. And it's essentially a laser that points into the ocean and looks at what comes back at very specific time intervals. And this LiDAR can penetrate into the ocean, and depending on the time at which the data returns and the strength of that return, you can say something about what's in the water. And so this is one of the sort of really novel, interesting technologies that was applied during the NAMES campaign. aside from the technology, there's just a project of the size just has a lot of people. I think we calculated in total there are about 278 participants, both science and logistics personnel, as well as ship personnel. You can see the list of the science party here in the center, and here's me, one of many in the middle with this black star. And so I was the ship logistics lead, which meant I really had to take on a large part of the project and deal with many different aspects of the project. And some of those, so I'll come back to that in a second. Some of those logistics include coordinating with the administration of the ship. And so each ship has a home base, and there are people that are associated with these ships whose jobs are to work on logistics for the ship. What I found out is scientists are not trained necessarily or funded to be logistics coordinators, but it's a big job that someone takes on. And so all this coordination, whether it comes to shipping, determining lab space on a ship, and having long conversations and meetings in person and over Zoom about what people need for sampling, what their water budgets are, because a CTD rosette is limited by the amount of water that's available. There's certain samples take priority over others just because they have to be sampled prior to others so that they don't get contaminated. There's scientific infrastructure on the ship that we have to make sure is in place or get so that it is in place, including things like fridges and freezers, liquid nitrogen for samples, and seawater systems. So like not only do we have a rosette to sample with, but we have specific diaphragm pumps that we pump water with to put through instruments and to sample from, like continuously underway while we're on the ship. And one thing that really has been a learning curve for me in my career is as I've taken on different roles in terms of logistics leads or as a chief scientist on a ship, you become an HR person. So human resources becomes important, and how you manage relationships, deal with issues that come up, really can make or break a project. And so it's important to, if you don't have those skills, acquire those skills if you think you're going to find yourself in those positions. And one thing we found in names was when we started was that it was such a big project, and there were people doing strictly ocean work, people doing ocean sea exchange work, air sea exchange work, people who are strictly working in the atmosphere, and people who do models of all three of those, is that we didn't speak the same language. We didn't even know what each group was measuring. In fact, some of those who weren't involved in the proposal writing project. And so we had to create diagrams like this that people could refer to and say, oh yeah, this group measures cloud condensation nuclei. And so it was a very big learning curve, not just for my own research, but to learn other people's research and figure out how those different processes might actually interact and what are the interesting research questions. You can write the research questions in the proposal, but once you're out there doing the research, you may find something really interesting that you think you can collaborate with that wasn't originally in the proposal and what was funded to be done. And like I said, quickly realized that organization was the key to keeping a large group of 200 plus people on task and understanding what the goals were as we moved forward. So the platforms, as I said, we had the C-130 that was a NASA plane that was essentially rented to the project. And then the Woods Hole RV Atlantis was the seagoing platform for the 4 cruises. So we had the same 2 platforms for all 4 cruises, which was also critical because once you set up all the logistics on a ship for a project, adapting those to meet another the specifications that another ship would offer, it would not be very straightforward. So we were fortunate to have both of these platforms throughout the entire 4 years of this project. So now I thought I would transition into a couple of examples of some of the work I did through the NAMES program. And as I showed earlier, the LiDAR from the aircraft looks into the water, and you can you get retrievals of different optical properties from that. And what we saw as in the figure here, the black line is the ship track. The yellow represents the sea level anomaly, so sea level height essentially. And these yellow circles are eddies in the North Atlantic. And the ship crossed this eddy. I'm sorry, can you guys see my pointer? Maybe that's better. And we crossed this eddy, and what we saw was some really different properties in the phytoplankton community. And the idea of the aircraft was that it would fly ahead or behind the ship, different days, different times potentially, but also survey the same waters that the ship was crossing. So in this figure, again, the thick black line is the ship track as it crossed this eddy. Here, the gray color, the lighter colors represent this eddy. The thin black line is the aircraft track at a different date that crossed at a similar location as the ship. And what we had to do was use some of the modelers that were part of the project to backtrack. If we seeded particles where the ship was, where was the plane at the same time or when it crossed that particle field? And so on the right-hand panel, what we're looking at is the back trajectory of particles or plankton. And so we can locate at what time that aircraft crossed through that same water mass. And so the yellow, we're not too concerned with here. This is an actual longer trajectory if we were to go back months and months. But what we're interested in the red was just about 2 weeks apart when these two tracks essentially overlapped the same water mass. And so what did we see when we crossed that water mass? On the left-hand panel, this is data from the LiDAR. So this is on the top panel is backscattering. The middle panel is essentially a measure of eccentricity or particle shape based on some of the other optical properties that are returned to the aircraft. And the bottom panel is chlorophyll to backscattering ratio, which is essentially a measure of the physiology of the phytoplankton in the water column. And you can see that between the two red arrows where you have the aircraft crossing this eddy, you have distinct changes in the optical properties that were being measured. We also saw in the ship's underway data, which is on the right-hand side, so this is the water that was being pumped through an optical system, also distinct changes when we went moving from the south to the north of that eddy. And we see these really distinct optical properties change over the course of that ship track as we went through the eddy. So what did we actually see in the eddy? This red box represents this discrete samples that were collected from the underway system in the eddy using an imaging flow cytometer. So this is a cytometer that analyzes cell by cell, but this one actually takes pictures. And so we can identify on the right-hand side, you can see the colors represent specific groups of phytoplankton. You can see that the community changed very distinctly as we went through that eddy. And in the bottom hand is some of my data set, which is using this BD Influx or the standard flow cytometer. We can look at the scattering properties and how those change when we're both south of the eddy, within the eddy, and north of the eddy. And those, if you look at, for example, and we can look at a couple of distinct phytoplankton groups. If we look at the side scattering properties, for all groups, they changed really dramatically. So it's likely that the scattering properties, not just the groups changing, but the scattering properties, which represent both the size and the density, or essentially carbon content of those cells, changed dramatically as we crossed through the eddy. And that's essentially what we were seeing. The LiDAR was representing, the LiDAR data was also seen from the aircraft. As I said, aerosols were an important part of the NAMES project, and how different aerosols affect how ice clouds are formed, or the ice within a cloud, and at what temperatures those clouds form, and how that would affect the albedo. It was an important component. And during our project, we saw that there was a 4-day period, and prior to that 4-day period, there was a storm-induced phytoplankton bloom. So we had a really deep mixing event where the mixed layer went from about 20 meters to over 200 meters. And in the 4 days following, we saw a phytoplankton bloom. And during that time, of course, we were sampling. I was sampling using the flow cytometer to look at different groups of phytoplankton. This picture in the upper, this upper picture here is actually a small pontoon boat that's actually we deployed while we were on station. And underneath that pontoon boat, there's air being pumped and bubbles being formed. And those bubbles produce aerosols, which are then essentially sucked through a hose and sampled so that people can look at their specific properties. So these aerosols from the seawater were being collected. Phytoplankton groups were being sorted. I sorted very specific phytoplankton groups, and we collaborated with Sarah Brooks and her student, Elise Wilborn, to look at how do phytoplankton impact ice nucleation of clouds through this aerosol or an aerosolization process over the course of a phytoplankton bloom. And so these are the results. And what Elise points out here in these key points is that the freezing temperature, essentially the ice nucleating temperature, freezing temperature actually rose throughout the course of this bloom from 28 to negative 24 degrees Celsius. And if we look at these three different specific groups and how the temperature at which they produce ice or affect the ice nucleating temperature, we can see that at about the salinity of 5 parts per thousand or 5 grams per liter, you get ice nucleating temperatures that are in that realm or in the range of what they were seeing through the aerosols being produced through the through this instrument they call the C-sweep. And so we also see that if more salt is added to the sample, you actually get a decrease in the nucleation temperature, which makes sense. As you add salt to roads, you change the temperature at which it freezes. But if we actually look at the salinity of down here of 5, which is actually what aerosols essentially are, is they extrude salt through the process of aerosolization, we have phytoplankton being able to actually impact the ice nucleating temperature of primary aerosols. So that was a really cool result. Another example of some interesting observations that we saw during this cruise, and I will play this video. This is Dr. Tom Bell, who's an aerosolic, studies aerosols and chemicals in the atmosphere and in exchange with seawater. We came across some of the extremely clean Arctic air in our study that was coming down from the north. And this sort of like made a lot of us realize how important aerosols are or how they impact our life. And so I'll play this video. Hopefully it will play here. And
so we have
so I guess the take home there is the next time you're drinking a cup of coffee and you see steam, you know that there's particles in the air, that it's not completely clean air. The reason you see the steam is because there are actually particles that are actually aggregating the water around them and becoming aerosols. Anyway, that was interesting. So this is sort of wrapping up the names results here, however brief they are today. And of course, everyone's got these after cruise pictures, but in reality, this is, for those of you who have done this at sea, know that these are really the pictures that capture sort of the work and the exhaustion that goes into being on a research cruise. I will say that NAMES is approaching 100 publications, and there's still a lot more that will come out of this project. And most publications from the Earth Venture program, these are the most from an Earth Venture program to date. Okay, so moving on to example 2, and again, I know we're sort of limited in time today, so I'll briefly go through what this expedition looks like, and maybe at least one research highlight from it. So NASA Exports was looking at the export processes in the ocean, so looking at how carbon gets from the surface ocean to depth. Unlike names that had a ship and an aircraft, there were two ships, a survey ship and a process ship. The process ship was a Lagrangian or water following ship, and so we tried to follow the same parcel of water and do research experiments to look at rates and things like production in the surface ocean. The survey ship ran different patterns around the process ship to actually look at what are the broader patterns around this parcel of water. And again, this is a large community-driven project funded by NASA with a budget of approximately $40 million. And this project is still ongoing, and we'll have actually a second sort of modeling component of it that will be funded next year, I believe. So as I said, the goal and rationale of exports is to predict the export fates and carbon cycle impacts of ocean primary production from satellites and other observations. It really took advantage of a lot of those autonomous assets like bio-Argo floats, sea gliders, and I have some examples of the data sets that come out of those different technologies. And the idea was that there would be two different community states. We have one in the North Pacific, which is sort of a low production, highly recycling microbial community in the surface ocean. And that's represented here by this food web on the left, where you have lots of different interactions in the surface ocean before anything is actually exported to the deep ocean or below the photic zone. In the North Atlantic, the idea was that we have a much shorter food web, and you have production going more directly to things like zooplankton, which are then excreting that material below the photic zone. And so we have these really different, this comparison and contrast between two different sort of sites. The Atlantic cruise was originally supposed to happen in 2020, but as we all know, there was a global pandemic, so it was postponed to 2021. This is what the PI list looks like, and this probably doesn't even cover it now because we've collected more PIs over time, and NSF has started to fund additional PIs to work on this project. But this gives you an example of how many people, and these are just the PIs. This doesn't include students or postdocs or undergraduate students who are working on the project. Outlined in red are what we call the project office. We're sort of in charge of determining how the logistics happen, how decisions are made. And I was fortunate to be part of that and was a chief scientist on both of the Pacific and Atlantic expeditions. Of course, it's not just pretty faces. Everyone's got a role, has a different sort of expertise, everything from robotics to chemical oceanography to physical oceanography, trace metal chemistry, microbial ecology, people who specialize in particle aggregation, disaggregation kinetics, a lot of different approaches that were being used to tackle these questions of how carbon is transported from the surface ocean to the deep ocean. To give you an example of some of the logistics of tracking samples, here's a short list of some of the discrete samples that were collected on the first exports campaign. Here's an example of my flow cytometry data, and this is just the surface of 40 meters. You can see over time, these are the samples collected for these different measurements and the sheer number of samples. And we can if we look at different discrete bins, you can see the sheer number of samples that are coming out of this project. And so having a program that really tracks these projects and when they're collected and how we integrate all these different measurements is really critical. This is an example of the different assets that were being deployed during exports. The yellow here is a sea glider. The red and purple coming in are the two different ships, and you can start to see the different ship tracks and the different assets being deployed. The green assets are sediment traps, both neutrally buoyant and those attached to surface drifters. You can see the ship trying to track the glider, which was a Lagrangian-based glider and drifter. And over time, you see different assets being deployed and recovered. And so this is essentially what we were seeing or what we were doing when we were in the North Pacific Ocean. And so obviously there's a lot of robotics and expertise in GIS and GPS tracking assets so that we can actually find them and recover them. And it's really talks or speaks to the nature of the sort of diverse level of expertise that's required in a project like this. So now I'm going to show you some examples of the data, this yellow glider that was deployed. We have the data from that glider. And this is, again, just one asset that was out there. There were a number of bio-Argo floats. If we get to the next screen here, we can start to see all of the different components that were collected. We have fluorescence or chlorophyll from fluorescence, the temperature profiles that were being collected by that asset, salinity, oxygen, and here's backscatter. This is a proxy for biomass or carbon. And then you put the temperature and salinity together to get the density gradients. And we can, from that, look at a broader scale than just a single platform. But considering this is a one platform, we have dozens, literally dozens of different platforms collecting similar data that we have to put into the same sort of frame of reference to understand the water masses that we were studying. So in the 2021 experiment, we actually added a third ship, not that the things weren't complicated enough. So we had 3 ships, all of these autonomous assets. And as I said, we had to deal with COVID. So we actually wrote up 78 pages of protocols. We had to quarantine for 2 weeks in a hotel prior to getting on the ship. And if I didn't have respect for those who had to quarantine or were alone during the pandemic, I certainly did after that quarantine. It is not something I recommend to anyone. The major differences between the Pacific and Atlantic, not just the food webs, but the Atlantic is a much more sort of turbulent and dynamic system. And again, because it's so turbulent and physically active, we were really trying to target one of these sort of eddies to put our assets in, so that we weren't trying to track these all over the North Atlantic, but a retentive eddy that would keep our assets within it, not only because it was easy to track our assets, but an eddy provides somewhat of a consolidated ecosystem to study, given that the eddy holds its shape and form for the time period that we're out there. There was a lot of work that went into identifying eddies, and I won't go into that today, but we were able to find one and study it. And again, there was the process ship, the survey ship, which actually surveyed outside of the eddy as well as inside, and then a third ship that we added that was out for about half of the same time and had different questions that were being asked. I'll skip that. Of course, we have to have a drone shot of all the ships that were out there when they were in the same water mass, and so this is footage from that drone. We can see how close the ships were. We got even closer. I think at one point we were within a couple hundred meters of each other, so that we could actually, do CTD casts and have confidence that we were collecting samples from the same depths, from the same water mass for comparison later. And that's important for calibrating across ships when we're thinking about different instruments that measure the same thing. We want to know that we actually have data that's compatible, and we know that we were measuring in the same data frame. Here's a nice picture of that one of the CTDs going in from the ships. You can see the other ships in the distance. And so it was really exciting to be a part of that and see that sort of work going on. Again, all of the work that happened during the program, I'll just point out quickly that some highlights from this process ship. This is the ship I was one of the chief scientists on. Some of the numbers here on the right just point out the massive amount of work that was going on. 2,000 samples for dissolved organic carbon, almost 2,000 chlorophyll extractions. Those are just the extracted chlorophylls. Those aren't even the high performance liquid chromatography data set that's coming out of it. Over 1,000 marine snow aggregates were caught and counted, and some of those were picked individually to look at the microbial communities that were on those aggregates. Anyway, just a lot of different aspects of the sampling and the sheer number of samples that occurred. And examples from the Sarmiento de Gamboa, which was our third ship that year. highlighted in red, 78 million liters of water were passed through a mock nest. So this is a opening, closing zooplankton net. Over 3 million images were captured by an instrument called a stingray, which images larger particles. Over 4 million images were captured by the flow cytobot, which is the imaging flow cytometer, which looks at the phytoplankton. So just massive amounts of data that are still being processed, and it will take some time to have all that data processed and in the right form to share with the public. One question that sort of came up during this last project, or the last Atlantic cruise for me was, if we look at the profile of fluorescence and forward scatter for one group, the Senecacoccus, in my flow cytometer, you can see distinct patterns with depth, and this is based on physiology and light availability. But one of the collaborations I had was with my friend Dr. Amy Moss, who's at Bermuda, who's also happened to be at URI when I was there, and we were fortunate to work on this project together. Was, can we use these profiles to understand zooplankton grazing to some degree? She's a zooplankton ecologist, and so her job for me was, can you give me some fecal pellets, and are there living phytoplankton within them? And there were. We then analyze those on the flow cytometer the same way I would for whole seawater. And then can we use this relationship to identify the depth at which those fecal pellets were formed? Right? And we can. It was really exciting. And so we can see that these different organisms were, at least the fecal pellets that we obtained from them, were that they were eating at different depths. And we can say with some degree of certainty what depth that fecal pellet was formed at. And this is important when we think about, were they feeding above or below the mixed layer? And what's the fate of that carbon that would have been in that fecal pellet had it been excreted at that same depth? And so this has got a lot of implications for modeling of carbon cycles using information from fecal pellets and zooplankton. And I know I'm running out of time, so I won't go into how we actually validated that, but we did. And there's a lot more data to come out of that project. Again, some cruise photos just to show that there's a lot of people involved, a lot of different technologies being applied. So my last example, and I'll keep this quick because I think I'm running out of time, is a pilot study called BioGoship. And GoShip is a program that has been going on for a number of decades. It was originally a different program by name, but essentially a global scale physical oceanography program. And in 2019, coming out of a SCORE working group in 2018 and 19, the idea was we need to put biology on these GoShip cruises to make a global data set of both biooptics and biological data sets, so that we can start to do these large scale global surveys of biology and look at changes over long term. And so there was a call in 2019 for statements of interest. Not knowing what would happen, I just submitted literally, I think it was 500 words, and I was selected for the team. And so this is the BioGoship team as it stands now. There are 5 of us that are participating in this project. We've written a proposal, and aside from that proposal, we were funded by NOAA for a pilot project. And these are an example of the science questions we're asking. Looking at the global patterns of taxonomic diversity, how those relate to the physical processes, and how elemental stoichiometry is reflected in the biology and chemistry that's being measured, or the physics and the chemistry that's being measured on these GoShip projects, and thinking about the different approaches that we're using to answer some of those questions. And here's a brief list of some of those measurements we want to collect. A lot of optics through the inline system, as well as some discrete samples for flow cytometry and imaging flow cytometry. Some CTD work on some cruises. We will have our own CTD so that we can do a biological specific cast. Other casts were sort of limited to what's available to us. And then a lot of samples that go back to be processed once we're back from the cruises. And this is a map of the GoShip lines. All these lines are different lines, and they're supported by different countries. These red dashed lines are the ones that BioGoShip will be participating in. We were actually went to this cruise off the coast of Africa, but due to COVID and some positive tests on the ship, that line had to be abandoned. Our next line starts up here, this P2 line, it'll actually be going from Guam to San Diego, and I'll be going to Guam in 2 weeks to help set up for that cruise. And then the hope is in 2023, we'll have our last cruise in the Indian Ocean, with the hopes that there'll be additional funding in the future to continue this program long-term, for a decade at least, so we can start to look at, are there long-term changes as we resurvey these lines over time. And with that, I will just say thanks. I know I think I'm out of time, and be happy to take any questions.
All right, at this time, I'm opening up for questions. So please speak to the mic very closely so it can pick up your voice. So who wants to ask the first question? Nobody? Stryovi.
Hello, thank you. Since most of your career has related to research, I was just wondering what advice you would have for an undergrad going into independent research. Like, what lessons have you learned in the beginning of your research, whether that's through education or your professional career?
Yeah, good question. And I did think about that a bit as I was putting this together, knowing the audience. And I mean, honestly, I would say that do participate in as many things as you can. I know it's not easy given like the time constraints people have, monetary constraints, whatever it may be, but whatever you can get your hands in, participate, and really do, in grad school for me, when I got there, I wasn't quite sure what I wanted to do. I had a good idea when I went in, and then it changed. And I just said, okay, well, let me just look around. And I started participating in cruises, really working on these different projects. And if you just put your heart a little bit into any project, it will pay out. There's so much need for people to do work to help out on different aspects of projects. I came out of grad school, I think with 2 to 3 publications out of my dissertation, honestly. 1 chapter and 2, maybe 3 papers, I think it was. But I had like 3 or 4 papers that had nothing to do with my dissertation. And honestly, that's, I think, what got me my postdoc position. They saw that I was productive, that I had participated in lots of sort of well-rounded research that wasn't highly dedicated to one aspect. That would be my suggestion is just participate in things. Be willing to volunteer, and it'll pay off.
So I know you said you've been at sea a lot. I was wondering, have you noticed like your time at sea has helped you get opportunities that you otherwise wouldn't have, and how were you, and if so, were you able to use that sea time to get those opportunities? And does it open up more positions for you on a boat as well when you go out for research cruises?
Yeah, good question. I think all of the research I presented to you today, with the exception of the LIDAR cross section of the eddy, those were not projects that I set out or research questions I set out to answer. And those came about by being on a research ship and talking to my colleagues about what was interesting. And I think it's those conversations. It's going to sea, for those who haven't done it, you have this, like, you're sort of, maybe less so today, but because of technology, but you sort of are isolated and you get to focus on what you're doing. And you're really sort of just like all in when you're there. And those pictures of people sleeping is true because everyone is so involved and just working. But through that work, you have lots of conversations about, hey, what are you working on? What's that? And that really spurned a lot of the opportunities for me for interesting research and publications that came out of it. As far as create opportunities down the road, I know for a fact that I was selected for that BioGoship team because of my participation in the names and exports program. They saw that I had volunteered to take on those logistics lead and chief scientist positions and said, here's someone, who has the experience of helping run a program. He has, at minimal, some background in research that's important to this project, but he's at least taken on these sort of larger project roles and knows how to get things done. And I'm pretty sure that opportunity came out because of my previous research at sea, my experiences at sea.
Hi, I was just wondering why you didn't want to do the deep sea benthic work, or why you didn't stick with it.
Yeah, good question. You know, I found it interesting. What I found were it's a pretty limited community, and not that they weren't accepting community, but the opportunities are much more limited. And so looking at what was available, and sort of, at least for me at the time and place where I was starting to do that work, the opportunities seemed a little bit limited. And it was also a little bit out of my background. I came out of a microzooplankton lab where I was used to looking at phytoplankton, used to looking at microzooplankton. And that's the sort of work I was doing and more familiar with. And honestly, it was what I was more interested in. I think I had convinced myself that the deep sea work was, you know, sexy. It was what I wanted to do. But when I got into it, it wasn't my field. It didn't feel right to me. And I looked around, the opportunities didn't seem to be there. Because especially if you want to, you know, there's only essentially one submarine you can go down in and collect samples. There are ROVs, but right there are dozens of research vessels and one submarine. So the opportunity to get onto those platforms is much more limited. And right, I don't think one research field is more important than the other. It was just what I was comfortable with and what I saw as a interesting future for myself.
All right, I think that's it for questions. Jason, thank you, and go Beavers.
Thanks, everybody. That's right. Thanks, guys. Appreciate it.